Hi everyone. Whatever time of day it is for you, welcome to our webinar, and it's going to be covering approaches to address the system qualification of the Simcyp Simulator. My name is Oliver Hatley. I'm a principal scientist at Simcyp Certara, and before I invite Karen to talk, I'm just going to go over a few housekeeping notes. I'm sure some of you will be wondering, yes, this session is being recorded, so if you missed a bit, you should be able to watch the recording back and listen again. And the slides will also be available on request. As you may have noticed, you're all on mute, but that doesn't mean you can't interact with us. So we've got QA facility, which is live throughout the duration of the webinar today. So please submit your questions, and we'll be taking questions and putting them to Karen at the end of this session. In terms of the rest of the agenda, over the next hour, we're going to be first hearing from Karen about PBPK modeling requirements, regulatory requirements. Then we'll stop for a quick poll, just to make sure you're paying attention, make sure you're not too distracted by emails. You can interact with us, have a few questions, and then finally we're going to cover approaches to address qualification, looking at the recent CP2 PSP publication, focusing on qualification of the Simcyp Simulator for CYP-mediated drug-drug interactions involving inhibition. Karen, I'm sure, needs no introduction to the people on the line. Previously, she was head of PBPK consulting services for 8 years. So she has plenty of experience interacting with clients and regulators. So no surprises, she's now the SVP of Client and Regulatory Strategy for Certara. Okay, Karen, I'll pass over to you.
Right, thank you very much, Ollie. First of all, can you hear me okay?
Yeah, perfect.
Okay, perfect. Right, let's try and move on to the next slide. Where's the problem? Okay, right. Okay, so before we move on and talk about the recent publication that we had on the qualification, what I wanted to do is just take a step back from that and talk about, what we mean by qualification. And so if we think about model-informed drug development, there's been a lot of discussion about it, a lot in the regulatory guidance talking about it. And one of the things that I wanted to point out was that, the FDA recently introduced their model-informed drug development pilot program. And one of the things that they mentioned as being new on there was, did you know that under the pilot program, they're asking the MIDD meeting requests and meeting packages to include elements of a credibility framework? And it's envisaged that this will facilitate alignment and streamline review. And so what I wanted to do was talk a little bit about that as well, because as you can imagine, when we're talking about MIDD and looking at the different modeling approaches that you have on the right-hand side, POP-PK, exposure response, PBPK systems pharmacology, when we're talking about applying MIDD in terms of context of use, it's important then that we have more streamlined or more documented processes to be able to actually address this. So when you're thinking about MIDD approaches in drug development, typically, in regulatory applications, they've been routinely used, for optimized dosing, provide evidence, supportive evidence for efficacy, informing clinical design, and guide regulatory policy. But what we're seeing more of is an increasing number of occasions when quantitative models have served as primary evidence, proving especially useful when clinical trials are not feasible or ethical. And this is something, certainly that I'm not going to speak to the other MIDD approaches, but what I am going to talk about is obviously PBPK modeling. And there's been an increasing impact of PBPK modeling on the label, especially when we're talking about clinical DDI studies. I'm not going to take you through all of this, but just to show that, these are increasing every week, every month. We're putting more on here, more instances of when PBPK modeling has been used to support the label and also in regulatory submissions. So now moving on to the credibility assessment framework, it's not really surprising that when you think of the number of submissions that are being done to the regulators globally, that is, in terms of the PBPK modeling, that we're seeing more of these types of documents and publications where we're being asked to consider a credibility assessment framework in MIDD and potential application to PBPK modeling and simulation. And this paper was published a couple of years ago. And I'm not going to take you through all of these steps, but just to let you know that one of the main areas of interest with respect to this credibility assessment framework is talking about context of use and defining the question to be addressed. Also looking at the impact of the model in terms of the influence and also the decision consequence that it's going to have. That you have to make an assessment of the model risk and then bearing all of that in mind, come up with a validation plan. And of course, this is actually based on the American Society of Mechanical Engineers, and it was for assessment of the credibility of computational models used for medical device applications. So what it's trying to do is streamline this approach. And as I mentioned in a slide couple of slides, sorry, in the slide couple of ago, that really what we're trying to do is streamline the regulatory approach. And so what I'm going to do is talk you through this over the next couple of slides and particularly focus on the context of use, which we're hearing a lot more about, and also in terms of defining the question to be addressed. So if we think about a typical scenario for CYP3A4 substrates, one of the main questions that we want to know is that if we've got an investigational drug, how should this drug be dosed when co-administered with CYP3A4 modulators? And this would be our question of interest. In terms of the context of use of the PBPK modeling, typically, we would have clinical data available with strong 3A4 modulators, inhibitors, and also inducers. And what we're trying to do is essentially use the PBPK modeling to inform on these DDI studies with weak and moderate 3A4 modulators. So we would be using simulation to predict the effects of weak and moderate 3A4 modulators on the investigational drug. And then this would be followed up. We'd be using these predictions to try and guide dosing recommendations in label. So we've defined the question, and then we've defined the context of use of the model and what we're actually wanting to use it for. So if we think about then the model development process for PBPK, we would have the model construction, we would have the in vitro data and whatever clinical PK data were available, we would develop a model. Then we would utilize our 2 clinical DDI studies if they were available. We had the itraconazole and rifampin studies. So we're talking about here typically a CYP3A4 substrate. And of course, what we would do then is verify the model or refine it if necessary so that we can capture the 2 clinical DDI studies. And then we would apply this model to untested clinical scenarios. So in other words, weak and moderate 3A4 inhibitors and inducers. And then what we would try and do is utilize the magnitude of interaction to extrapolate to a dose adjustment. And of course, this is the typical model development process that's been referenced as best practice and been mentioned time and time again in a number of the global regulatory guidance documents. But when we think of the regulatory assessment, what I talked about in the previous slide was developing that model for the investigational drug. and then going through that process. And typically what we would do is prepare a report describing the data that we utilized, describing the model development process, the verification steps, and all of the steps that were actually involved in the application as well. And also included in that, we would perhaps discuss sensitivity analysis, the context of the data, and any uncertainty associated with it. But this is all with respect to confidence in the drug model itself. After that, we would then be wanting to know what is the confidence in the context of use. So in other words, we've taken that model, we've applied it now to these untested scenarios where we're looking at the effect of weak and moderate inhibitors or inducers within the platform. So we're utilizing a combination of the compound files and the algorithms within the platform to try and capture data with other substrates. So we've got the drug model itself, but then we've got the confidence in the context of use or the intended use. And therefore, we would be expected to provide additional supporting documentation in the form of perhaps compound file summaries. So within the Simcyp Simulator, we have documentation for each of the compound files that we have in there, indicating the source of the data and also verifying each of them against the different substrates or inhibitors, depending on whether we're talking about an inhibitor or a substrate. And then what we could do is, in support of that context of use, provide a short report providing the level of confidence that we have when utilizing these weak, moderate inhibitors and inducers within the platform. But also, in addition to these individual reports, there are many publications out there in the literature as well where the Simcyp Simulator has been used for 3A4, for many other enzymes and many other scenarios. And so I suppose really when we're talking about providing confidence in the context of use, we could be talking about, as I said, publications that are in the public domain, collating the reports and also providing the compound file summaries. So there's the report for the confidence in the drug model and then providing that additional support itself. And really, I think this is what I want to focus on in the second session here. It's how do we actually address this? Because it's not stipulated in any of the documents. It's indicated that you've got to provide, supporting documentation for the context of use, but there aren't clear indications in any of the guidances about the level of support required. And in fact, in some cases, it appears that the level of supporting documentation appears to be associated with regulatory impact. So if we look at the EMA guidance on the reporting of PBPK modeling and simulation, what they indicated very clearly was that it depends on the regulatory impact of the simulation. So if you look at this schematic here, we've got low, medium, and high regulatory impact. And obviously, when we're talking about high regulatory impact, what we're talking about here is potentially replacing clinical studies or looking at non-studied scenarios or reinforcing sparse data. At the bottom of this, you can see that if the model is going to be used for in-house decision-making only, it's got low regulatory impact, and therefore the level of qualification or verification is actually relatively small. So on top of the actual documentation in the form of publications and also individual reports and compound file summaries that I've talked about so far, what the EMA have indicated is that what they would like to see is a formal qualification process that they would have the opportunity to review. And when we're talking about qualification, just an example here, if you wanted to know or quantify the effects of an investigational drug being a victim of a drug interaction, so in other words, if you've got a CYP3A4 drug and you want to you've run simulations with a weak or moderate CYP3A4 inducers and inhibitors, and you want to waive clinical studies, then it's expected that you submit a supporting level of documentation. And what they've indicated here, that in order to qualify the ability of a PBPK platform to predict the effect of inhibition on a specific enzyme, then what we would need is a pre-specified qualification data set. And this should, if possible, consist of a series of drug substances. So you've got victims and perpetrators. And what they want to see are victim drugs, that eliminated to variable extents through the metabolism, through the enzyme of interest. So in this case, we're talking about CYP3A4. So before we actually then go on and move on to the second part of the process, what we want to do is we've got a few questions that we'd be very interested, to get your answers on, especially based on the previous slides that I've just presented, and also your experiences with respect to qualification and regulators. So what I'm going to do now is just hand over to Ollie, who's going to take you through these questions.
Thanks, Karen, for that first part. So if we bring up the first one, so the first question of the three is based on your experience, or we can also think about your perception if you don't have that much experience. Have the regulatory requirements for supporting documentation for PBPK models changed over the past 5 years? And yeah, what we're quite interested in is if there's any difference between FDA or EMA, for example, or yeah, if you think they've both increased or stayed the same. So we'll let that run for a little bit just to give you a chance to reply to that one.
Yeah, so as Ollie said, I mean, one of the reasons that we're interested in this really is because, there being an increasing number of publications, I think, coming out from the FDA, as we mentioned, the credibility framework. The EMA, they've made it very quite clear about what their position is on the qualification. And therefore, we're just trying to, collate this information based on your experiences or perceptions, really, as Ollie's indicated.
Okay. Looks generally like both have increased. Okay, that's interesting to know. We've got another poll question now. It's a bit about understanding. So do you understand the documentation required for regulatory submissions involving PBPK modeling? Again, in this case, what we're interested in, yes, no, or maybe you don't have experience, so you can put not applicable.
And, I think this might seem like quite a basic question, but it just goes back to the beginning part of my presentation and why we're interested in it. Because, as we've indicated, there are several different types of documentation, and it's not always clear, I think, the extent of documentation required for the different regulators. So it's just trying to gauge that level of understanding.
Right. Okay. Across yes and no, it's kind of 50-50, isn't it? For that one. Okay, thanks for that one. And in terms of the last one, so we're obviously talking about inhibition in this case, but which of the following would you prioritize in terms of qualification and context of use for PBPK modeling? So induction, transporter-mediated drug-drug interactions, or organ impairment?
I mean, obviously, there are many more areas of interest, but, these are three that we keep coming back to and keep hearing a lot about. And therefore, we'd just be keen to know what your priorities are in terms of, application of PBPK modeling.
Mm-hmm. All right, shall we give that a few more seconds? Okay, quite a split. So yeah, around a third each, really, for induction, organ impairment, and transporter-mediated DDIs. Okay, thank you, everyone. I appreciate you doing that. And what we're going to do now is move on to the second part, as we discussed at the beginning, and look at this case study in terms of the publication, in terms of approaching and addressing qualification. So I'll hand back to Karen for that.
Okay, thank you very much. So yeah, well, thank you very much for taking part in that poll, and I think especially that last question as well, because as you're going to see in the next few slides, there's quite a lot of work involved, and I think certainly looking at the approaches, that we've been using, and so it was just interesting to gauge what the priorities are likely to be going forward. So obviously, in terms of the qualification areas. We've got, CYP inhibition itself, which can consist of competitive and also mechanism-based inhibition. There's CYP induction alone or in combination with mechanism-based inhibition. We've got transporters, the OATs, the OATP1B1s, PGP, BCRP. And in terms of the CYP enzymes as well, we've got a number of different CYP enzymes in terms of inhibition and induction that we have to consider. Special populations, we've been doing a lot of work on pediatrics, organ impairment, pregnancy, certainly over the last few years, and been a lot of emerging publications on the, the applications here. But I think these are just some of the key areas. I mean, we haven't even mentioned UGTs or any other, phase 2 enzymes here. But what we're trying to say here is that when we're thinking of qualification, what we have to be thinking about are the mechanisms, the enzymes, and the different populations. And therefore, it's important, I think, when we're thinking of the approach, that we think of them in terms of these areas and how we're going to move things forward. So in terms of the actual qualification approaches, obviously what we have to do is identify and collate these examples. We've mentioned several times that there are lots of publications on the application of the Simcyp Simulator from many of the consortium members, different research groups. There is a lot of ongoing work. And it takes time to identify and collate these examples because they may have been performed in different versions. And so it's a case of pulling all of these together. What we can do as well is, perhaps run simulations ourselves, try and pull all of this data together and submit as a single publication on a single mechanism or a single enzyme. Also, as we've mentioned previously, that sometimes when submitting the PBPK reports for specific investigational drugs to regulators, they've been submitted alongside short reports describing the context of use and providing supporting data in the form of compound file summaries, but also independent analyses for these. And of course, what we can do as well is go for formal qualification with the EMA. So one of the things that we decided to do was actually start with the qualification and in terms of CYP inhibition. And what we focused on was CYP1A2, CYP2C8, CYP2C9, 2C19, 2D6, and 3A. And we looked at competitive inhibition and mechanism-based inhibition. Now, obviously, at the time of doing this, we could have gone to the literature, had a look to see what was there, collated all of this, talked about the different versions that were available. But this is extremely time-consuming, valuable, but time-consuming. And then how do you actually put this together in a single report that can actually be used to qualify a specific mechanism or a specific CYP enzyme? So what we decided to do was actually do an analysis ourselves. And if you think back to one of the earlier slides that I presented, what we talked about was the fact that a qualification database was required, and we're referring to it here as a qualification DDI matrix. And so what we did, and I'm using the royal we here because of course it was all my colleagues at Simcyp who did this. What they did was look in the University of Washington Drug Interaction Database for all clinical DDIs involving these following enzymes here. And this alone was a huge amount of work, but we felt it was extremely valuable to do. Because typically what happens is if we submit a PBPK report, the question that comes back is, well, provide qualifying documentation. And we felt that it was important for us to know for each of these enzymes, how many clinical DDI studies are there actually out there, and to have all of this information collated together. And then what we did was focus on those DDIs where we had compound files available within the simulator. So that was the initial focus. And then after that, what we did was, in terms of look for the FDA recommended reference substrates and also inhibitors. But what we'd done previously anyways, most of the compound files that we have within the Simcyp Simulator include these FDA recommended reference substrates anyway. But also we felt it important that we try and capture, inhibitors and so weak, moderate, and strong inhibitors and substrates that were susceptible to differing degrees of inhibition. So once we'd done all of that analysis, then we went through and had a look at all of these and sorted them according to these criteria here. At the time as well, if there were any inhibitors or substrates that we identified, what we tried to do was a feasibility analysis to see whether it was possible to actually to develop a compound file for that. Because obviously for some of these enzymes where we may not have many clinical studies, it was important to try and increase this by especially if we could, add a substrate or an inhibitor, to boost the number of clinical DDI studies overall. So it was quite a laborious process. And what we ended up with was 123 clinical studies involving competitive inhibition. So CYP1A2, CYP2C8, and you can see here that not surprisingly for CYP3A4, we've got 62 studies available. And for CYP2C8, 4 studies, and for CYP2C19, 4. And again, and so we had a lot of discussion at the time, sort of saying, okay, well, should we just go with CYP3A4 because we've got a lot of studies there, and CYP2D6? But at the end of the day, these are the studies that were available. The substrates that are available are pretty robust. And also, if you look at the CYP competitive inhibition mechanism, for most of these enzymes, it's similar across the board. And therefore, what we decided to do is go for these 5 CYP enzymes here. 6 CYP enzymes. In terms of mechanism-based inhibition, we found a total of 78 studies. Again, not surprising, 52 CYP3A4. And in terms of 2C8, slightly more. So these were the studies that were carried through. Workspaces were prepared for all of these, and these simulations were run using version 19. Now, again, I keep coming back to the DDI matrix, and this is important because, if we want to have a high level of confidence in this, it's important for us to have a range of different substrates for a range of different inhibitors, and that essentially these compound files are being cross-verified. So in other words, what we don't want to end up with is a midazolam substrate that's only been verified with ketoconazole. What we've got is a midazolam substrate that's been verified against cimetidine, cyclosporin, fluconazole, fluvoxamine, itraconazole, and ketoconazole. And that's what we're trying to show here, the fact that each of these substrates, each of these inhibitors has been cross-verified, and that is why we're referring to it as a DDI matrix. So this is the DDI matrix for competitive inhibition and also for mechanism-based inhibition. And what we're showing on the right-hand side here are just whether they're clinically sensitive substrates, moderate or moderate, strong, weak inhibitors. And obviously the size of the box is indicative of the number of clinical DDI studies. Importantly, if you think back to the qualification criteria that the EMA indicated, they said that it would be nice to have a good spread of drug substances. And so what we did as well within the publication is we indicated what the FM values were for each of the substrate, what the bioavailabilities were, and also the FG and FH values. And importantly, if we're thinking about CYP3A4 substrates, ideally to have the ideal qualification data set, what we would want is to have a spread like this, so that if we've got a substrate that is metabolized by 50% by 3A4 and a low to moderate bioavailability, that it would actually be captured by this data set that we're seeing here. And on the left-hand side, you can see that what we've got are the substrates and their FM values for the other respective enzymes. And in terms of the CYP inhibitors, there were about 24 inhibitors involved in the analysis. I'm not showing all of them here, but what I'm showing you are the type of data that we actually provided in the supplementary table. And it's important, I think, just to talk you through this, because typically for the CYP inhibitors, when we're developing the compound files, what we try and do is obviously have the files as mechanistic as possible. But sometimes for these inhibitors, if we don't know how they're metabolized, then we might have them represented by their PK data. Then we would put in the in vitro inhibition characteristics, verify that we can capture the observed exposures, and then run the simulations against substrates. What we're indicating here is whether the Ki values, they've been corrected for binding, but whether they were optimized or used on the basis of in vitro data. And for these Ki values and also the inactivation parameters, a meta-analysis was done for each of them based on all of the data that were available in the literature, corrected for the non-specific microsomal binding. And these are the substrates as they stand in the version 19. And of course, what we tried to do as well was capture the, whether there were strong or moderate or weak inhibitors. So I've got a couple of slides here. What I don't want to do is talk you through all of the results. What I'm trying to show you here are the elements of the information that we've provided within the paper and how that can help you when determining, qualification and context of use. So what we've done is we've represented the data in terms of changes in Cmax and AUC ratio according to the different mechanisms. So this is what we can see in this particular plot here. Graphs A and B refer to competitive inhibition. C and D refer to mechanism-based inhibition. And we've got these sorted according to CYP enzyme. In the table next to it, what we've done is also indicated what the average fold error is, the bias and the precision with respect to competitive inhibition, mechanism-based inhibition, and each of the individual CYP enzymes. Also, what we've done as well for the competitive inhibition, we've indicated by substrate, indicated by inhibitor, and also we've provided the same level of information with respect to mechanism-based inhibition here. And this is so that if there was a specific drug that you were interested in, we've got the granularity here that allows you to look at a specific drug-drug interaction. So in terms of the context of use, the main reason why we separated all of those according to that level of detail was because if you've got an investigational drug and you're wanting to focus on competitive inhibition or mechanism-based inhibition, those data are there. If you're looking at a specific CYP enzyme, those data are there. If you're interested in looking at the mechanism, the CYP enzyme by substrate and inhibitor, then we have those data available as well. And as I said, this is really important because it allows discussion of the qualification data set for context of use. And so in other words, if we're talking about, if we've got a CYP3A4 drug, I'm going back to the initial example, and what we're trying to do is say that what we want to do is a wave clinical studies involving weak and moderate CYP3A4 inhibitors. We've got our report submitted for our specific investigational drug, and what we could do is then use the information from this data set here in terms of context of use and say that we've provided evidence here showing that the CYP3A4 weak and moderate inhibitors perform well against the compound files described in this analysis. So I think when also it's important when we do these types of analysis that we make it clear what were the key findings and what can we actually provide in terms of guidance? Well, obviously, the main point of the paper was to run all of the simulations for those 6 enzymes for mechanism-based inhibition and also competitive inhibition and say, fantastic, we've got substrates and inhibitors that predict very well. And therefore, if you've got an investigational drug, which you're looking at as a victim drug, these are the guidances that we can provide. So for a number of the substrates included in the DDI qualification matrix, a clinical DDI study was used to optimize their FM values. I mean, obviously, what we do initially is we take the in vitro data, we would run a simulation, determine whether we could capture the DDI, the clinical DDI. If not, we would refine that FM value to show that we can actually capture that. And or we would utilize, the mass balance data along with in vitro metabolism data to actually refine the relative contributions and the clearance routes. So at this point, what we are saying is that for most of the substrates that we have within the database, we used a clinical DDI study or mass balance data to actually refine the clearance mechanisms. And therefore, if you want to use this as a qualification data set, what we're saying is that if you had an investigational drug and you were able to demonstrate using clinical DDI data, then this would act as a qualification data set. But this is quite typical anyway. This is what we tend to find with CYP3A4 substrates, that we tend to use a clinical DDI study to refine it. So there's nothing new here, but just to make it clear what we're talking about in terms of context of use and qualification data set. So now this to me is the more interesting aspect, because this is often what we find is early on in drug development, we might know what the inhibitory potency of a drug is, but not necessarily know how it's metabolized and all the different clearance routes involved. And so what we wanted to do was try and break this down. What we found is that of the 18 CYP3A4 inhibitors used in the study, only 3 of them had optimized Ki values. So what we're trying to do is provide guidance here. So if you've got an investigational drug that inhibits CYP3A4, then what we are saying is that what we can do is, if you've got a positive control Ki value that is also in our data set here, what you can do is essentially calibrate that. Or if it's similar, if the Ki values for the positive controls are quite similar, then we can say that we have confidence in your Ki, in vitro determined Ki value, and run a simulation with that. And without doing any sensitivity analysis, and that you're probably more likely to be confident in that. But obviously, if you flag a positive or a clinically significant DDI, then you would want to do a sensitivity analysis just to sort of see what range you're actually falling into. And of course, this comes on to the next slide here, where what we're talking about are the other enzymes. So among the 30% of inhibitors, so 37% of the inhibitors, an optimized Ki value was used. And the difference between the optimized and in vitro values was about tenfold. So what we're saying here is that if you've got an investigational drug and you want to assess the DDI liability, that we would recommend dropping the Ki value by up to about tenfold and doing a sensitivity analysis here. And that this qualification data set can be used to actually support this, because this was one of the findings that we've got from actually doing this analysis. So this was obviously just some of the key aspects. I haven't even talked about the mechanism-based inhibition, but within the paper itself, we've also indicated important steps that if you've got an investigational drug that's a mechanism-based inhibitor, what the recommendations would be. What we're trying to show you in that publication is the level of detail, that we've provided. I haven't even mentioned the fact that for each of those inhibitors and each of those substrates, we have the supporting compound file summaries in version 19, all of which can be actually, contribute towards a formal qualification process. So what we've decided to do is with respect to CYP inhibition and competitive and MBI is obviously we've collated all of these examples. We've done a formal analysis, shown that the compound files within version 19 perform well. And so what we're going to do now is we're actually going to submit this work as for formal EMA qualification. So we've got the publication just to fast track and make those data available to everyone. But now we're going to go through the formal qualification, which can obviously take about up to a year. But obviously what we have to think about are the other areas. What about CYP induction? You know, obviously in the poll that we conducted earlier, this is why we were interested to know your thoughts. There's CYP induction. And again, we consider it alone or in combination with mechanism-based inhibition. There are transporters, special populations, pediatrics, organ impairment, all of these areas that you indicated an interest in. So what we're going to do, and what we have been doing, is going through the same process again that we've gone through for the CYP inhibition, where we identify studies involving CYP induction. We also collate examples from the literature that we can use in the meantime until we've done this supporting analysis. And at some point in the future, we can go for a formal EMA qualification. But obviously, we have to make sure that we have enough relevant examples in order to be able to do that. So in the meantime, what can we do with respect to special populations? Well, I think it's important, again, I know I keep going on about it, but context of use, that is what is really important. And if you think back to the EMA slide where they're saying that the regulatory impact will dictate the level of qualification necessary. Obviously, if we're wanting clinical trial waivers in special populations, we have to have a reasonable amount of data to be able to support that in terms of its qualification. And obviously, there have been ongoing efforts over the last few years. And if we want to get regulatory adoption, we have to, what is good about this is the IQ publication that came out a couple of years ago, where they looked at PBPK modeling in renal and hepatic impairment. And the good thing about this was that there was a nice cross-section of drugs available from each of the industry members who contributed to this analysis. Also, from our own in-house consultancy case studies, we can continue to apply the models and gain experience there and submit them to the regulators as well, just to gauge their level of interest and to share these data. And also, where possible, we've been publishing this data as well to put it in the public domain. But on top of this as well, we recognize that there may be gaps, and therefore we have to continue advancing the science. And that is just indicated in this particular publication here. So just to quote, I think one of the FDA presenters from last week from the ISISX webinar, I thought it was quite interesting there because they were giving their position on where we are with respect to organ impairment and regulatory submissions. And I remember from one of the slides, I just saw verification, verification, verification. And I think, that is essentially what I'm saying here, is that there are emerging examples, independent analyses, and therefore we have to keep these coming out, but we also have to think about the best way of actually utilizing this data going forward if we want to be able to qualify the platform. And also, I think importantly as well, if we're wanting to get regulatory adoption as well, you'll be aware that the renal impairment guidance has been undergoing review and been circulated to everyone. And therefore, I think it's important to think about, based on the data that are available in the public domain, what are the approaches that we can use? What are we more confident in? And this was just a publication that a colleague and I put together, and just thought process as a perspective in CPT, where we were thinking that, based on our experience, we've seen a number of the, examples or case studies where we've got data available in severe renal impairment or severe hepatic impairment. And if you have those data available to verify the, the model, then perhaps we can extrapolate to untested scenarios, perhaps in mild renal impairment, and also following multiple dosing as well. So this was just a schematic that we put together. And I think, these types of analyses are important going forward, and especially when we're wanting to look at all of the different special populations and thinking about the best way that we can actually, provide support for regulatory submissions. So without further ado, I just wanted to acknowledge, apart from the individuals who were on the publication, the Simcyp Qualification publication, I just wanted to acknowledge the efforts of the Simcyp team, because this is not just based on work from 6 individuals, but from many years of work, and I just wanted to acknowledge their contribution. So I'd like to thank you very much for your for your interest and attendance. And I know that qualification isn't the most exciting topic available, but we felt it important to share the findings of our study, and also to indicate that we are, trying to move things forward in this particular area. Thank you very much.
Thanks, Karen. So we're going to move into a period of questions. So just a reminder, you can use the Q&A functionality to send your questions through. I'll kick us off with some questions. Why do we need qualification if submissions are already approved?
So I think that's a really interesting question, and in fact a very important one, and and it's something that really I should have addressed during the presentation. But just to go back to the CYP3A4 example, or CYP DDIs, I think many people would say, why are we bothering to qualify competitive inhibition for CYP enzymes when there are so many examples out there? And I think the issue that we have here is that, depending on which regulator it goes to, and within the EMA, obviously, there are a number of different agencies, and you could get, a very different outcome depending on where it ended up. And in terms of the formal qualification, what we're talking about here is having a standardized approach. And I think this is where the EMA are going with this. So in other words, if we submit qualification for CYP DDIs for competitive inhibition across the board, then at least it will take away that subjective, I think, viewpoint with respect to looking at the PBPK models, and hopefully a more streamlined process, and yes, and more transparent as well.
Yeah, makes sense. Thanks. Just a note, because you mentioned in the presentation that you were using version 19. Obviously, that's a few versions now. We're on version 21 in the simulator. So is regulatory qualification version specific? Do you have to do every time?
So again, yes, I did mention during the, webinar that, the publication that we did, all of the work was done in version 19, because of course that's what we had available at the time when the work was initiated. And of course, this is quite typical of, many of the submissions done with a consortium. They would have started with the version that was available when they did the initial model development. And so I think going forward, to answer your question, the qualification is version specific, but what we have been doing, and what I've seen by some of the regulators, is that even if you provide supporting qualification for a different version to that was used for the investigational drug simulations, I think as long as you can show version control and that there's been little change across the versions, and you provide supporting documentation indicating that, say for example the compound file summaries, then it can be extrapolated to other versions, the findings. Yeah.
Okay. Yeah, we had a similar question as well, basically saying, if you had to go through it with every version, does it really pay off? Anyway, so another question is about complex DDIs with PBPK qualification, with more than one mechanism for inhibition or induction for perpetrators, and especially for substrates. So yeah, can you comment on the process involved for qualification in these circumstances?
Yes, so again, it goes back to, finding relevant examples. So, obviously I've simplified it somewhat by talking about the processes independently, but I think we're collecting enough examples now of where we've got transporters. We've also got UGTs and also CYP enzymes. And for those examples, it will be important to utilize, I think, published data or data that have been made available in the FDA reviews, collate those examples, and essentially put them together in a single document to support those. But we are seeing an increasing number of examples. But again, going back to the point that I was trying to make earlier, I don't know what the magic number is in terms of the amount of number of examples for qualification, because I've seen in some cases where for CYP3A4 we've had a lot of supporting data. I've also seen instances where there've only been a handful of examples. So, again, there are a lot of different examples out there, but it's a case of pulling them together. And that's why, as I said, I think it's interesting in terms of, it was interesting for us to see in terms of priorities.
Yeah. I think this kind of leads on to what you're just saying in terms of priorities, but obviously we've got a long history of using PBPK for CYPs, especially as we've talked about today, interactions, and lots of data for verification. But given the large data gaps, are we really ready for qualification, for example, in special populations?
So, again, I go back to the point that I made, about the FDA presenter from last week in the ISSX workshop, verification, verification, verification. I think, and it comes back to, again, what level of qualification we want to, we provide, because obviously, when you do submissions to the, to the FDA, they are asking for, qualification or examples showing that, the platform works sufficiently well in those areas. And that was what was mentioned with respect to special populations. So it may be that we don't have enough data, I think, to go for formal qualification and endorse the platform across the board for the special populations. But I think, there are enough examples available in the literature or emerging examples that can be collated and put together in the form of a document.
Okay, great. Related to populations, there's a question here about if we change some parameters in populations, for example, gastric emptying time, should we re-verify Simcyp default compound files, midazolam, itraconazole, etc.?
So I would recommend looking at doing that, yes. If you're using those compound files for the support in that particular population. So I'm assuming that what you mean there is that if you change, say, for example, you were looking at renal impairment and you'd run a simulation and you decided to change the gastric emptying there, would you then do the same for the other? Yes, I would. I would look at both scenarios. Yeah.
Okay, great. There's a comment here about the 10-fold sensitivity analysis for Ki for a competitive inhibitor. The comment is, it seems like it's a lot if you're working with high quality in vitro data. Was there any consideration that the 10-fold adjustment needed in some of the PBPK models for inhibitors came out of lack of solid in vitro data across various compounds, or a good understanding of FUMIC?
So, it's always, so that's a great question, and one that we've, the analysis we've taken apart many times over, because at the end of the day, when you're doing a meta-analysis, what is the best way of doing it? Do you put all of the data available there, correct for non-specific microsomal binding, and then just take an average Ki value? Or do you streamline them, look at them to see which ones, certainly in my view, would have robust methodology and had corrected and used low protein concentrations? And so, we've had a look at all of that. And so you say that 10-fold seems quite high. The problem that we have is obviously across the enzymes, when you look at the spread of the in vitro Ki values, in some cases it's much higher than that. And that is why what we've done is recommended that, or tried to say for CYP3A4, you know, that we're pretty confident that the in vitro Ki values hold, but then we've provided the criteria that, you know, your positive control Ki value should be similar to one that we've used in the, you know, the analysis that we provided. And I think, you know, for the other CYP enzymes, probably what we could do going forward, as we increase the number of examples that we have, we would be able to provide similar guidance for that. But at the moment, the tenfold is the best that we can do based on that that data.
Yeah. Okay. Thanks. So in terms of access to post-marketing surveillance data to verify PBPK models, do we have access to that? For example, if the model predicts no DDI and the clinical trials are waived, do we see the same in the real world clinical data? Maybe do you have any examples of
that? No, I don't. I don't have any examples of that I can share with you today, but I think, you know, this is the point really that I'm trying to make, you know, during this session today, is that there are a lot of data out there. It's a case of deciding, you know, what, you know, so there are several things here that we need to think about. There are a lot of data available, but what type of data do we need, and what is the best data that is actually going to help us qualify the platform going forward? And so I think that's, you know, a very key point. But I think if people do have examples that they can share with us, then it can only, you know, help us, you know, build better qualification examples and reports for submissions.
Mm-hmm. Good. I'll just quickly go back to the tenfold Ki question, if that's all right. And it was just a clarification on whether it does tenfold Ki for 3A apply to both gut and liver? Was FU gut assumed to be 1 in the analysis? I guess in the first question, yes, it's applied across the board, but I mean, the FU gut will be an interesting one.
Yes, and so what we tried to do was look at that with respect to FU gut, and of course what we have in Simcyp will be a mix of FU guts depending on the actual recovery. So at the moment it wouldn't be possible for me to answer that question, but what we can do is go back and do a sub-analysis and provide that information. Yeah.
Mm-hmm. So there's a comment here from Neil Parrott, and he says there are 3 possible applications: induction, transporter, and hepatic impairment. I guess that was the organ impairment options that we gave in the polls. The hepatic impairment might have the biggest impact, but faces big challenges given the complexity. Do we have the right data and understanding of hepatic pathophysiology to make progress at the moment, or is a more long-term effort necessary? And if any plans at Simcyp for such work?
Oh, well, I'm going to bat that one to you, Ollie.
Yeah. So, well, yeah, I can say for organ impairment, that is something that, as Karen's mentioned, that we're starting to do. In terms of making progress with the hepatic impairment populations, we obviously made some strides, I think, with version 21 and trying to incorporate other features within the model. But I think even from what was presented even last week at ISSX, it was really interesting talk by Tycho talking about the impact on absorption and bile salts and things. So there's still lots of work to do for the hepatic impairment populations, I would say, but it's definitely something that we are continually working on.
Yes, I think just to add to that, you know, I think also what came out of the ISSX, you know, workshop presentation last week was that at the moment, I think the level of the confidence in hepatic impairment models is such that they would request clinical studies in each of the, you know, mild, moderate, and severe. But I think, there's still the utility for PBPK going forward and, for multiple dosing and also potentially DDIs as well. And so that goes back to my point is that I think even if we feel that we're at the position where we can't avoid clinical studies, we still have to then provide supporting documentation, I think, to support all of these other scenarios.
So there's quite a lot of interest in the KI, and there's one more question here. Have you observed in your analysis any bias by system, i.e. KI from HLMs or recombinant SIPs?
No.
No. Okay. So see if there's any more questions. I think we're about at time anyway. Yeah, if there's any more questions that come in, we'll try and answer those ones. But I think in the interest of time, and it's hit top of the hour, I think we should close this. But before we go, just to mention that there is an upcoming webinar on the 22nd of June. This is from Emi Kimoto and Athena Varma, and they're looking at OATP1B mediated DDIs, and also quite a hot topic at the moment, looking at biomarker-informed model-based risk assessment. So please register for that one on the Simcyp members area if you're able to do that. And then finally, I just wanted to again thank Karen for your time and giving the presentation today. So thank you for that, and thank you for everyone for joining online. Okay, bye everyone.